Hello and welcome everyone. In last class, we learnt about DNA demethylation. We understood all of the possible mechanisms which a cell can adapt to undergo DNA demethylation. So in those different pathways, we learnt about how the direct removal of methyl group is not possible because of breaking the carbon-carbon bond, which is quite stable. It is not thermodynamically favorable, and that is why this event, direct removal of CH3 group, has this possibility. Other mechanisms were involved DNA repair machinery. So the DNA repair machinery involving mechanisms were base excision repair pathway and nucleotide excision repair pathway. In the base excision repair pathway, the 5-methyl cytosine, which undergoes deamination, and it gets converted into thymine. On this thymine, the TDG, thymine DNA glycosylase enzyme works, and this thymine is thymine and T and G mismatch is repaired and replaced by the new cytosine. This is how in the base excision repair pathway DNA demethylates. While in the nucleotide excision repair pathway, the whole patch of DNA in which there are unmethylated cytosines, this is removed and new nucleotides are added. While there was other mechanism which was newly discovered due to the discovery of TET proteins, 10-11 translocation enzymes, which has TET1, TET2, TET3, all those proteins, all those enzymes are responsible for the oxidization of 5-methyl cytosine. So let's take a look at the whole process of what DNA demethylation. So let's start from DNA methylation itself. So cytosine on the CpG dinucleotides is converted to 5-methyl cytosine. Okay, and this reaction is mediated by DNMT, DNA methyltransferase enzymes. The CH3 group donor is SAM, S-adenosyl methionine, which gets converted into S-adenosine homocysteine. So this is the reaction. This is what we call it as DNA methylation. So this 5-methyl cytosine can be further converted. This can be further converted, means this can be further oxidized to form 5-hydroxymethyl cytosine, and this reaction is carried out by TET enzyme. So it can be carried out by TET1, 2, or 3. Okay, all those enzymes are responsible for conversion of 5-methyl cytosines to 5-hydroxymethyl cytosine, in which oxygen is converted into carbon dioxide, while the alpha ketoglutarate, the substrate, is converted into succinate. So this 5-hydroxymethyl cytosine again can be further converted into 5-formyl cytosine, and this 5-formyl cytosine can be further oxidized into 5-carboxyl cytosine. Again, this steps are TET mediated. All of those require oxygen as well as alpha ketoglutarate as the substrate. Oxygen gets converted to CO2 while alpha ketoglutarate gets converted into succinate. So this is the whole, this is the DNA methylation. Well, from 5-methyl cytosine to the different series, the different products, 5-hydroxymethyl cytosine, 5-formyl cytosine, and 5-carboxyl cytosine. All of those demarcate DNA demethylation. This is what we have learned till now. So this 5-hydroxymethyl cytosine, 5-formyl cytosine, and 5-carboxyl cytosine, these are the markers of demethylation of DNA. So let's take a look at different TET proteins. So so this is the schematic of the structure of the TET proteins. So this is the TET1 protein, which is 2036 amino acid long. This is TET2 protein, which is 1912 amino acid long, while TET3 protein, it is 1680 8 amino acids long. Okay, so all three proteins have different amino acid lengths, but the overall structure is somewhat similar. Here you can see this red domain is present in TET1 and TET3, while it is absent in TET2. Again, the structural similarity can be found in this domain. This whole green, orange, and blue part, this is constant in all of those. Okay, so let's take a look at what are those domains, what are those different domains. So this red domain is known as CXXC domain, and this is the domain which is responsible for binding to CpG islands. So this due to this domain, the TET protein is recruited on the CpG islands. So the green domain is cis-rich domain, cysteine-rich domain, and it is very important for oxidation of 5-methyl cytosine. So 5-methyl cytosine is converted into 5-hydroxymethyl cytosine. So for this conversion, for this oxidation, cysteine-rich domain is crucial, while the DSBH, double-stranded double-stranded beta helix domain is essential for the binding of metals. So different metals, most probably iron, could work as the cofactor of the whole reaction, and for binding of those metals, this domain, double-stranded beta helix domain. Let me write it here: double-stranded beta helix domain. This is required. So beta helix, double-stranded beta helix domain is for binding of the metals, while this rich domain is for the oxidation of 5-methyl cytosine. So just when you take a look at the roles of those domains, you can see that though if those domains are absent in the TET proteins cannot carry out the DNA demethylation, and that's why those two proteins are carrying out the catalytic activity of the enzyme, and that's why those are known as catalytic domain. Okay, so as the TET proteins are going to catalyze, they are going to oxidize 5-methyl cytosine to different forms of the different oxidized form. So this catalytic domain is very important, and it should be present in all of those three, because of course this is governing the function of the TET protein, and that is why you can see this cis-rich domain, cysteine-rich domain, and this double-stranded beta helix domain. This is common in all three proteins. So this is common in all three proteins. While the CpG binding domain is only present in TET1 and TET3. It's absent in TET2. While this orange region is spacer region, the function of spacer region as well as this gray region, which is known as PRK12323, those are unknown. So the functions of spacer region as well as this domain, these are unknown. So we haven't discovered those yet. So this is the basic structure of TET protein. I hope all of this is clear to you. And let's meet in the next video lecture, and we'll discuss about the active demethylation versus passive demethylation. Till then, see you. Bye bye.